Okay, welcome everyone. Good afternoon to you. Hope you're doing okay. Uh, First thing, if you can hear me okay through the internet, would you please type yes into the chat window? Okay, wonderful. Thank you very much. Okay, so I hope you're doing well. It's good to see that Ian is in front of the Leaning Tower of Pisa at this time. Okay, so take this time off from school to do a little virtual traveling. All right. Gang, I have a question for you. I put into the chat window. Are you aware of any local companies that are offering free internet access at the home for right now during our coronavirus emergency? The reason I ask is because I'm starting to have students that are saying that they're not able to afford their internet access because their work hours at work have been cut. So they can't afford the internet, which means they can't continue their course, and it's starting to become a real problem. Have you heard of any local companies that are offering free access at this time? Okay, John, thank you. I'll have to look into that and see if there's something we can do for students, because we're headed for a real academic train wreck if most of our students, or at least some of our students, lose their internet access. Okay, well, gang, welcome back. Let's see here what we can accomplish today. Let me remind you, of course, that your Chapter 7 textbook homework is due by the end of today. Also, keep in mind that Lecture Exam 2, which is going to be next Thursday, so a week from today, April 9th. And that study guide for that exam has already been sent out. So if you have questions or concerns, please let me know. And just keep in the back of your mind at this time that the second literature review 2, is due on April 21st. The article for the second literature review, if you have not looked at it yet, is considerably longer than the first article. So you definitely don't want to wait to the last second to start working on that. Okay, before we get started, do you have any questions at this time about what's happening? Okay, all right, gang. Well then, let's get started. We're going to dig deep into Chapter 10, talking about translation. In order to get the conversation started, what I'm going to do is start with a video, which gives you a nice graphical illustration of the process. The video is a little bit on the long side. It's about 7 minutes long, which is a little bit longer than I normally like to show in class. But I think because it's a very excellent video and it really does a nice job of highlighting important features of the process, I am going to show it to you at this time. Please expect that I may stop the video every minute or two to see if you're with me, to see if you have any questions. Okay, so bear with me a moment. We're going to get our introductory video on translation started. Okay, so let's see here. Here we go, gang.
To produce a functional protein, the genetic information found in the form of an mRNA, which is composed of nucleotide building blocks, must be translated into the language of proteins, which are composed of amino acids. This translation process is orchestrated by a large macromolecular ribonucleoprotein machine in the cell, known as the ribosome. It uses a key adapter species known as a transfer RNA or tRNA. tRNAs are the molecules that actually translate the nucleotide sequence into an amino acid sequence. tRNAs are bifunctional in nature, with 3 nucleotides poised at one end of the molecule to read the mRNA template in an anti-parallel fashion. This is referred to as the anticodon end of the tRNA. At the other end of the molecule is the acceptor end, where an activated amino acid is attached. This amino acid corresponds specifically to the identity of the codon being read. Aminoacyl tRNA synthetase enzymes are responsible for catalyzing the loading of the amino acid onto the appropriate tRNA species. The fidelity of this step is extremely high, which is critical to the overall fidelity of protein synthesis. In its most basic form, during translation, the tRNAs walk along an mRNA in the 5' to 3' direction. As they move, the tRNAs sequentially recognize or decode consecutive 3 nucleotide codons that correspond to a given amino acid. This highly orchestrated march along the mRNA template takes place in the heart of the ribosome, facilitated at each step by portions of this machine, as well as by extracellular factors that participate in each step during the translational cycle. The ribosome has binding sites for 3 different tRNAs. One tRNA is always bound in the centralmost position in the ribosome and carries the growing polypeptide chain. We refer to this tRNA as the peptile tRNA. It's bound in the P site of the ribosome. The incoming tRNA with the new amino acid to be incorporated binds adjacent to the P site in a site referred to as the A site for aminoacyl tRNA. There is a third binding site, the E or exit site, where the tRNA binds on its way out of the ribosome following the incorporation of its amino acid into the polypeptide chain. As in most processes, there is a beginning, a middle, and an end. The initiation phase of translation consists of the small subunit of the ribosome working in concert with various initiation.
Gang, do you have any questions at this moment? Are there any parts of that first 2 minutes and 40 seconds that you want to ask a question about? Okay. Not hearing anything, let's continue.
And an end. The initiation phase of translation consists of the small subunit of the ribosome working in concert with various initiation factors to identify the starting AUG methionine codon that is positioned in the P site of the ribosome. Next, the initiator tRNA engages the AUG codon and the large subunit of the ribosome joins in a catalyzed reaction. Because of the substantial differences in the mRNA structure between bacteria and eukaryotes, this process is quite different in these two systems. In bacteria, a polypurine motif in the mRNA, known as the Shine-Dalgarno motif, is found 6 to 8 nucleotides upstream of the initiator AUG. This sequence directly pairs with a polypyrimidine motif in the small ribosome subunit rRNA to position the AUG in the P site, where the initiator tRNA binds to it. In eukaryotes, the ribosome engages the 5' capped end of the mRNA and, in association with a collection of initiation factors, scans along the mRNA until it encounters the 5' most AUG codon. Because the initiator tRNA is part of this scanning complex, it plays a key role in finding the AUG and positioning the ribosome complex where it belongs. After initiation is complete, the ribosome is ready for iterated rounds of amino acid addition. This phase, referred to as elongation, can be broken down into 3 basic steps. In the first step, the ribosome must select the aminoacyl tRNA that corresponds to the codon poised in the A site of the ribosome. This step is facilitated by the GTPase elongation factor EFTU in bacteria or EEF1A in eukaryotes. Simply put, the process of tRNA selection consists of 2 phases. In the initial selection phase, an aminoacyl tRNA in complex with EFTU can be rejected or accepted by the ribosome. Once the complex has been accepted, GTP is hydrolyzed and EFTU leaves the ribosome. A near cognate tRNA can still be rejected in a second phase, which is referred to as proofreading. In both steps, thermodynamic and kinetic mechanisms are used by the ribosome to aid in discrimination such that the system chooses, with extremely high fidelity, the cognate tRNA from the 30 or 40 tRNAs in the cell. Once the tRNA selection step has taken place, peptide bond formation is catalyzed in the large subunit. In this step, the growing polypeptide chain is transferred from the peptidyl tRNA to the nucleophilic amine of the A site tRNA amino acid. This transfer extends the polypeptide chain by one amino acid. At this stage, the extended peptide is attached to the tRNA in the A site. This whole mRNA-tRNA complex must now be repositioned in the ribosome to open up the A site for the next incoming aminoacyl tRNA. This translocation step is catalyzed by another translational GTPase, EFG in bacteria and EEF2 in eukaryotes. The GTPase couples the chemical energy of GTP hydrolysis to mechanical motion. These core events of elongation, tRNA selection, peptide bond formation and translocation happen iteratively until the ribosome reaches a codon in the A site that signals stop. Stop codons are recognized by protein factors known as release factors that function in an analogous manner to tRNA. One end of the release factor recognizes stop codons positioned in the A site with high specificity, and the other end facilitates chemistry in the active site of a large subunit. In bacteria, there are two release factors, RF1 and RF2, that recognize the three stop codons, UAA, UGA, and UAG, with overlapping specificity. In eukaryotes, there is a single factor, ERF1, that recognizes all three stop codons. The reaction that is catalyzed is a hydrolytic one, wherein the growing polypeptide is released from the peptidyl tRNA, resulting in the termination of protein synthesis. The final step in protein synthesis is referred to as recycling. This step promotes the dissociation of the large and small subunits of the ribosome and the release of the remaining mRNA and tRNA components. At this point, the small subunit is free to re-engage the initiation factors that allow it to begin again the task of identifying AUG codons to start another round of protein synthesis.
Okay, fantastic. Gang, so in my opinion, that's a pretty superb video. We would not have been able to show you a video like that 20 years ago because we didn't have all this crystal structure of the ribosome available to create an animation like this. So it really is amazing what we've learned in as much as 20 years. So what we're going to do today is walk through the finer points of translation that the video was trying to share with you. Do you have any questions about the video at this time? I will share a link to that video in my afternoon email to you. So any questions before we begin? Okay. All right then, let's get to a Microsoft Word document. We will continue our conversation about translation. As is normal for me, I will be emphasizing the prokaryotic information and just making some comments at the end about eukaryotic translation. Okay, so this is Biology 215, lecture on Chapter 10. Today's date is April 2nd. Okay, so this is going to be an overview of the translation process in prokaryotic cells. And here we go. All right, so for translation initiation to occur in prokaryotes, there's several factors that need to be in place, right? There's several puzzle pieces, if you will, that the cell has to have ready to go and have access to. All right, so translation initiation requires, obviously, that there be a piece of messenger RNA present. So messenger RNA has to be there. Next, we need a charged initiator tRNA, which in prokaryotes is a very special form of tRNA called an F-met molecule. The formal chemical definition of this or description of this is N-formyl methionine. So we need a charged initiator tRNA called N-formyl methionine, or F-met for short. Okay. Notice that this initiator tRNA, which is able to insert a methionine amino acid at the starting point for a polypeptide to be made, is a different tRNA than would be used to insert methionines in the internal region of a polypeptide. Okay. Okay. This initiator tRNA that can insert a methionine amino acid at the beginning of a new polypeptide is different and distinct from the average methionine tRNA used to insert methionines internal to the polypeptide, or at least after the very first position. Okay. Thirdly, to initiate translation, magnesium ions need to be present. Magnesium plus 2 ions must be present. And it appears to be the case that the purpose of the magnesium is to maintain the proper conformation, that is to say, the proper shape of the ribosomal proteins. Okay, ribosomal proteins are often abbreviated R proteins. If I start moving too fast for you, please let me know in the chat window. If you have questions, please type them in or unmute your microphone and speak up. Okay. So a fourth thing that needs to be present for initiation, translation initiation to occur is in fact GTP, guanosine triphosphate, which of course you know is a chemical cousin to ATP. GTP does definitely play a role in the translation process because it's an energy-rich molecule, and you did see in the video that there are moments where GTP is cleaved, and that releases energy to allow the ribosome to do things. But recent research is starting to suggest that GTP might have a role in the translation process in a way that's beyond or in addition to its energy capacity. It may be a signaling molecule which is involved in decoupled protein receptor signaling in cells, which if you've studied any anatomy and physiology, hopefully you've covered some ideas with decoupled protein receptors. So at any rate, GTP is definitely an energy source and may also have other roles involved in cellular communication. And then finally, as the video highlighted and which your textbook highlights, let me see here where that's at. Yes, table 1 in your textbook. Table 1 in your textbook, page 273, is a very nice organization of important ideas and molecules that are involved in translation in E. coli. And E. coli is typically used as the prototypical example of prokaryotic translation. The truth of the matter is that there may be a whole bunch of bacteria on the planet that do things differently than E. coli, but science has pretty much learned what it's learned from studying E. coli. So there's 3 proteins called initiation factors. And I will refer you to table 14.1 on page 273 of the current edition of the textbook. Okay, so these 5 bullet points: the messenger RNA, the initiator tRNA, magnesium ions, GTP, and 3 initiation factor proteins all have to be available to successfully allow translation to begin. Number 2, let me pause a moment as you folks get your notes completed. Okay. During initiation, the initiation factors, the IF factors, the initiation factors bind to the 30S subunit, that's the small subunit of the ribosome. Okay. The initiation factors bind to the small subunit of the ribosome, and that facilitates binding of the small subunit to the messenger RNA. Okay, so that's an important point, right? Ribosomes, the large subunit and the small subunit and the large subunit, they don't come together first and then thread the messenger RNA through the interior channel. That's not how it works. The small subunit actually attaches to the messenger RNA first, and then when that has completed, then the large subunit locks into position on top of that. Okay. Note how the complete ribosome does not bind to the messenger RNA. The small subunit binds first, followed by the large subunit. Okay. Now, binding of the small subunit to the messenger RNA occurs at a very special sequence in the prokaryotic messenger RNA that we referred to before and which was in the video, called the Shine-Dalgarno sequence. Okay. Coming out of undergraduate school, one of the first projects I was given by my mentor was to clone and express a protein. And I struggled for a month to do that. And the reason I struggled is because I didn't appreciate what a Shine-Dalgarno sequence was when I first started my project. So it really is the devil in the detail kind of things, where you need to know these bits of information if you're going to successfully do any genetic engineering. So called the Shine-Dalgarno, and this Shine-Dalgarno sequence is upstream of the AUG start codon. Okay. The Shine-Delgarno sequence, its consensus sequence, at least in E. coli, is the following. Okay. You take note in the video did not give you the consensus sequence. It simply said it was a purine-rich sequence. Well, there you have it. You can see all the A's and the G's. AGG is the consensus sequence, at least in E. coli. This consensus sequence is typically found about 6 to 8 nucleotides upstream of the start codon. Okay. Folks, are you okay? Am I going too fast? All right. Let me know if I'm going too fast, typing too fast. Okay. So continuing here with this idea of initiation, all right? IF1, initiation factor 1, is actually going to block the A site. So let's see here. Okay. Initiation is kind of cartoon diagram for you on figure 6 of page 274 of your textbook. So maybe page 274 would be useful to have open at this time while we're talking about things. Okay, the way initiation begins here is that IF1 blocks the A site of the ribosome. Okay, so that the very first amino acid, which is going to be the formulated methionine, docks into the P site. Okay, so that's an important thing to realize. Normally, during standard elongation, when a polypeptide is being made, new amino acids start at the A site, but that is not true for initiation. For initiation, the first amino acid is lodged into the P site because initiation factor 1, a protein, is physically blocking the A site. Okay. IF2 facilitates the docking of FMET into the P site. And IF3 actually prevents the large subunit from docking prematurely. We need to be sure that the P site has acquired the FMET amino acid first before the large subunit of the ribosome tries to sit down. If it tries to sit down before that, translation is not going to work properly. So IF3 prevents premature binding of the large subunit. Okay. If we did get premature binding, we may not get any translation occurring, or if we did get translation occurring, it would probably be a scenario where we have the polypeptide being made in the wrong reading frame. So this is extremely important that the large subunit is prevented from docking prematurely. Okay, so if premature binding is allowed, you're either not going to get any useful translation, or you're going to get a polypeptide that has the wrong sequence because the messenger RNA is translated in the wrong reading frame. Okay, so questions so far?
So tell me if you can't hear me, but if we were talking earlier yesterday about how most of RNA is not even translated, is the presence of a Shine Delgarno sequence what determines if it ends up being a sequence that's translated?
It's a very good, reasonable question, John. And I would say that that is frequently true. Yes. So if you were a computer jock doing a sort of statistical analysis on bacterial transcripts that are capable of being translated, you would find a high percentage of the total pool of transcripts that can be translated as having that Shine-Dalgarno sequence available. Yes. Other questions? Okay. All right, gang. Number 3 here. We're just walking through the translation process. Now, binding of the large subunit to the small subunit is going to require that IF3 leave. Okay, so once the proper formulated methionine amino acid has properly docked into the P site, the ribosome will eject IF3, and then that will allow the large subunit to come on top of the small subunit in the proper register, in the proper position, if you will. Once the initiator F-met amino acid is correctly placed in the P site, the small subunit ejects IF3, which then allows the large subunit to properly dock on top of the messenger RNA and small subunit. All right, so at this point, we have basically completed initiation. Initiation is all about the small subunit finding the messenger RNA and properly loading the initiator amino acid into the P site. Once that has occurred, we can now transition into elongation. Okay, so translation initiation ends and elongation begins. All right. Now, what's going to happen next for elongation to really ratchet up and do its activity in an iterative cyclical fashion is that a second charged tRNA is going to bind to the A site with the help of what's called an elongation factor. It's just a specialized protein that Mother Nature has created to facilitate the elongation process. The video spoke about it, your textbook spoke about it, and I'm going to put it in your notes. This special elongation factor is in fact called EFTU. Okay. A second charged tRNA binds to the A site with the help of elongation factor TU, or EFTU for short. Okay. The purpose of EFTU seems to be to increase the fidelity, meaning the accuracy of the translation process by slowing the process down sufficiently well that we don't get to mistakes being made by the ribosome trying to do its work too rapidly. Think about how many times in life when you've been trying to do something too rapidly, when you felt uncomfortable because you were rushed. Typically, that's when we make mistakes. That's exactly the same thing for the molecular machines inside our cells. If they try to do their work too quickly, they typically make mistakes. Okay, so let me see here. Okay, EFTU. Okay, EF stands for elongation factor, and TU stands for thermo unstable. Okay, that's, these things are named by the researchers that have been studying the processes. So when the researcher discovered this and realized that the purpose of this protein is to enhance the fidelity of the process, at some point in their thought process, they decided they were going to name this elongation factor thermo unstable. Okay. EFTU has been shown to increase the fidelity, the accuracy of translation by intentionally slowing the process down. Okay. Second part, stage B of elongation. Okay. Well, at this point, a second amino acid has been put into the A site. So that means we're now positioned. Okay, we have the initiator formulated methionine in the P site, and we have the next amino acid, whatever that may be, which is specified by the codon of the messenger RNA. We have that in the A site. So the very next thing that has to happen is we actually need to form the peptide bond. The peptide bond between the initiator methionine in the P site and whatever amino acid is in the A site. So a peptide bond is formed between the formal methionine amino acid in the P site and whatever amino acid is now in the A site. Okay, you know that the specificity of whatever amino acid is going to be determined by the codon of the messenger RNA and the anticodon of the tRNA that locks into that position. Okay, so that peptide bond formation, we covered the chemistry of the peptide bond formation at the end of last class. That's going to happen through the activity of 23S ribosomal RNA in bacteria, and the equivalent ribosomal RNA in a eukaryote is 28S. Okay, 23S ribosomal RNA is responsible for the creation of the peptide bond in prokaryotes. Okay, I'll just drop that down and tell you that it's 28S ribosomal RNA in eukaryotes. Okay, so let me see here. Let me see here. Okay, gang, I want to slow down just a moment and be sure that you're with me here. So I have a question for you. And I hope you have access to your textbook, because I really need you to have that book with you when we're talking. So the question comes from figure 7, So please take a look at figure 7 in the textbook. Okay. If you have the current edition of the textbook, the customized book that I recommend, that would be on page 275. So here's my question. Take a look at figure 7, take a look at cartoon step number 4 and cartoon step number 5. Take a look at cartoon steps 4 and 5. Okay. And I know it's very tiny. It's even tiny for me to look at it sitting right in front of it here. But when you look at cartoon step number 4, you see the transfer RNA carrying the polypeptide in the P site, and there are 2 amino acids there. Can you please type into the chat window what the color of those amino acids are? Okay, thank you, John. Purple and orange. Look very carefully. If you're missing it, they are very tiny. So look really into that image. They are purple and orange. So now I know you're looking at the right place. And you notice that there is an amino acid in the A site, which is colored, I believe, sort of like a light green color. So now that I know we're looking at the right location of cartoon steps 4 and 5, my question is this. As the ribosome creates the peptide bond, does the ribosome transfer the polypeptide in the P site to the amino acid in the A site, or does it transfer the amino acid in the A site to the polypeptide at the P site? I know that's a lot of words. Let me write that down on screen for you. Okay. Looking at the formation of the peptide bond, does the polypeptide at the P site get transferred to the amino acid at the A site, or does the amino acid at the A site get transferred to the polypeptide at the P site? What do you think, folks? Okay, so Kevin says A to P, and Morgan says P to A. Ian says P to A. All right, how about the rest of you? Okay, all right, it looks like the evidence is oh, John says A to P. Okay, all right, it looks like we're almost split. I would love to have you folks on a tug of war team. Look very carefully, because in fact, the ribosome is transferring the polypeptide in the P site to the A site, P to A. How do you know that? Because look at the color of the polypeptide sequence at step 5. If the amino acid at the A site were being transferred to the polypeptide at the P site, you would have purple, pink, and green. But you don't have that. What you do have is green, purple, and pink. Do you see that? Okay, so the answer is that the ribosome is transferring the polypeptide to the amino acid at the A site. Okay. All right, so that's our answer. And again, my proof is, look at the color of the amino acids, okay? Notice that at step 5, the polypeptide contains green, purple, pink. Okay? The only way that that could be the case is if the purple and pink from the polypeptide had been shifted to the green amino acid at the A site, and then that entire new polypeptide was ratcheted over to the P site. Folks, am I making sense? Do you have any questions? Okay. Seriously, gang, we can pause on this and talk if you need to ask a question.
So does this have to do with the whole attaching from 5' to the 3' end?
Let me think about your question, what you're asking me here. I've never thought about it in that way, John, but I think that would be reasonable. The whole 5' to 3' business is actually the next part that we're going to talk about, where we appreciate that the ribosome is actually going to pull, it's actually going to physically drag the messenger RNA through the internal channels of the ribosome in a 5' to 3' direction. In other words, after the ribosome transferred the polypeptide in the P site to the A site, in the very next moment, it pulls that fragment of polypeptide into the P site by dragging the messenger RNA from 5 to 3. So I think your question is related to this topic, yes. Other questions? All right. So, you really want to bear down on the details of figures 6, 7, and 8 for your upcoming exam, that you can really understand what's really happening here. Okay. All right. So, any further questions? All right. So at this point, we formed the peptide bond. We shifted the polypeptide from the P site to the A site. And now, in the very next step, the entire messenger RNA, tRNA polypeptide complex, which is momentarily in the A site, is going to be pulled through the ribosome a distance of 3 nucleotides, or 1 codon at a time. This is referred to as translocation. This is how the polypeptide that momentarily spends time in the A site is now shifted into the P site. So now we can repeat this entire process again. Load a new amino acid into the A site, shift the polypeptide to it, and then shift that entire complex back into the P site. All right, so how to put that in words? The entire messenger RNA, tRNA, polypeptide complex is pulled through the ribosome, 3 nucleotides, meaning 1 codon at a time. This shifting process, if you will, this pooling activity, if you will, is referred to as translocation. Now, translocation as a process hopefully it's starting to make some sense what we're talking about here. Translocation as a process is facilitated by another elongation factor called elongation factor G. Okay, the function of elongation factor G at this point is to actually to cleave a molecule of GTP and provide energy to the process. Translocation is facilitated by EFG, whose function it is to break GTP down to release energy. All right. So let's see here. So I'm just looking back in my notes a moment. This entire process we have described under number 4, starting with bullet point A. So A, B, C, and D. All of this, steps A, B, C, and D of note number 4, is going to repeat itself in a cyclical fashion until the ribosome encounters a stop codon. Okay. Repeated activity of steps 4A through 4D occurs until the ribosome encounters a stop codon. Okay? So, you know, some of the things I commonly find with students, and it's not a bash, it's just a reality that this is complex stuff, is that, you know, we've been covering, you know, the content of the course that we have covered and that you'll be tested on next week includes DNA replication, transcription, translation, all these different things like Okazaki fragments and origins of replication and promoters and terminators and Shine-Dalgarnos and stop codons. All of these terms start to get confused in your head and you start thinking about a stop codon being important to transcription. It's not. Stop codon's not important to DNA replication. You really want to be sure that you cleanly organize this knowledge in your own brain so that you don't confuse words with wrong processes. Just be very careful as you're studying. Okay, so steps 4A to 4D are going to repeat in a cyclical fashion until a stop codon is in fact found. Any questions before we carry out translation termination? Okay, gang. All right. So number 5, translation termination. Translation termination, the process of terminating the process begins when the ribosome encounters a stop codon. Okay, so let's step through a couple things with this. So the ribosome is going to encounter a stop codon. You know from the genetic code table that there's 3 of them. You don't have to memorize them. I will provide them on the exam. Okay. But the reality of it is, if you go into any form of genetic engineering down the road, you're going to be using this so much that you're just simply going to memorize it because you're using it so frequently. So the ribosome encounters a stop codon, and they are UAG, UAA, and UGA. Okay, and for these codons, there are no transfer RNAs which have anticodons to match them. Okay, there are no tRNAs that can bind to these stop codons. The cell doesn't have them. The cell does not have any tRNAs with an anticodon that can lock down onto the stop codon sequences. Okay, so that basically means the A site is going to remain empty in terms of any amino acids or tRNAs. There's simply nothing there in terms of a tRNA that can bind. And so what happens is that because the A site stays unbound for an unusually long period of time. Now, in the case, frankly, in the case of a real cell, you know, how long is too long? Well, we're talking a second. A second is too long here. So if that A site remains unbound for a second or longer, the ribosome is going to actually stall. It's going to be like you flood the carburetor of your car engine. It's going to stall. Because the A site is empty, the ribosome stalls. Okay. When the ribosome stalls, release factor 1 is able to enter the A site because there's nothing there. There's no tRNA there. Because the A site is empty, the ribosome stalls. This provides sufficient time for release factor 1 to enter the A site. Okay, so we have a protein that has a shape that allows it to nestle into an empty A site. Okay, the shape of RF1 allows it to bind into an empty A site. Okay. Now, when that happens, the purpose of the release factor 1, as was highlighted in the video, and which is highlighted on page 276 of your textbook, figure 8, is to in fact cleave the covalent bond between the polypeptide and the ribosome structure itself. In other words, the polypeptide that's in the P site, it's being held in position in that location by a covalent bond to the surrounding RNA and protein of the ribosome itself. So to release that polypeptide so it can completely move away from the ribosome, that covalent bond has to be cleaved. That's the job of release factor 1. RF1 breaks the covalent bond holding the polypeptide to the ribosome, which allows the polypeptide to be released. Okay. At that point, when the polypeptide is in fact released, that completely destabilizes the structure of the ribosome so that it falls off the messenger RNA. Release of the polypeptide from the ribosome further destabilizes the structure of the ribosome so that it falls off the messenger RNA. Okay, and it was at this point at the end of the video where the video talked about ribosome recycling and the idea that the ribosome, which is now fallen off this original piece of messenger RNA, could potentially reassemble around a different transcript that also needs to be translated. Okay. The released ribosomal subunits are now recycled in the sense that they are free to bind to new transcripts, to bind to new pieces of messenger RNA to translate them. Okay. All right, gang. I think that pretty well covers it. Do you have any questions at this time?
So I do have a question regarding the last point we just made here. So is there a certain amount of time that a ribosome can be used for this process, or are we making new ribosomes on a frequent basis? And if so, what is that time frame?
Okay. We are making new ribosomes constantly, but that rate varies tremendously depending on the type of cell we're talking about and what its physiological job is. For example, in your stomach right now, a lot of the cells lining your stomach are in fact dying because of the acid in your stomach. So the stomach has the highest rate of mitosis outside of your bone marrow. In the next 60 seconds, you're going to make 500,000 new cells. So to support the reproduction and physiological activity of these new cells, there's a very high rate of ribosome manufacture and assembly. But in cells like neurons that are not breaking down and turning over on a daily basis, we're making ribosomes, but at a much slower pace. Now, that being said, ribosomes can be recycled just like you can recycle objects at your house and reuse them. You might have a favorite shirt that you've washed 100 times and it's starting to look a little ratty, starting to have some holes in it. Ribosomes and any machinery in the cell is like that over a period, I would say, of probably, I'm ballparking this one, I would say anywhere from about 50 to 100 translations per ribosome. The machinery of the ribosome gets a little frayed and it gets to the point that it needs to be formally recycled, where all the ribosomal proteins have to be chewed up by the proteasomes of the cell. The individual amino acids have to be recycled so that new ribosomal proteins can be made. In a similar fashion, the ribosomal RNA is going to be chewed up in the lysosomes. The individual RNA nucleotides are going to be spit back out, and the cell can use those RNA nucleotides to make new ribosomal RNA to build brand new ribosomal subunits. So it's a complicated answer to your question. I would say the average ribosome can probably translate 50 to 100 messenger RNAs before it gets too worn out to do its job properly, and the cell will have to completely turn it over and build a ribosome from scratch.
Thank you, that's helpful.
Okay. Other questions, gang?
So can we recycle the mRNA similarly to the ribosome? Like, does this process damage that transcript to where it can't be used again to create another polypeptide chain?
Good question, John. Slowly over time, the transcript will be damaged. It may be damaged by the ribosome itself, or it may be damaged because there are moments of time where it's not being bound to a ribosome. While the messenger RNA is bound to the ribosome, it's largely protected from any other cellular enzymes that might want to chew it up. But in those moments of time where it's not bound to a ribosome, it's exposed. And so there will be some periods of time where the messenger RNA is about to be translated for the 27th time, but before the 27th ribosome can dock to it, some form of endonuclease in the cell goes over and nibbles on it and breaks it down. So the messenger RNA can be recycled, but it does also have a finite lifetime. Other questions, gang? Okay, good stuff. Good stuff. Feel free to keep asking the questions, type in the chat window, that type of thing. We don't have too much time left here. I want to try to get into your notes some of these important distinctions and differences between prokaryotic and eukaryotic translation. Notable differences in eukaryotic translation. Oh, what the hell did I just do? Oh, don't tell me I did something really bad there. Oh, well, okay, I can't undo that. Oh, I created a new window. How about that? Okay, sorry folks, slip of the finger there. Okay, let's keep going. All right, so that remember first, of course, that in eukaryotes, transcription and translation are uncoupled. What two processes did we mention last time are coupled in eukaryotic cells? Go ahead and type that in the chat window if you remember. Transcription and translation are uncoupled. Do you remember? Transcription and what are coupled? Okay.
That's the DNA processing. I have it in my notes.
Okay, yes, it would not be DNA processing. It would be
sorry.
RNA
Yeah,
processing.
you're right.
Yes, okay. So transcription and RNA processing are coupled. All right, number two, the presence, okay, eukaryotic messenger RNAs have this thing called a 5' cap. I've already noted that before, but that is a distinction between prokaryotic messenger RNA and eukaryotic messenger RNA. So the presence of the 5' cap, which again is formally 7-methylguanosine on eukaryotic messenger RNAs, is required for efficient translation initiation. Okay, so in prokaryotic cells, using prokaryotic translation, what you need for translation initiation to be efficient is you need a Shine-Delgarno sequence. That's not the case in eukaryotic translation. What you need is a 5' cap on the 5' end of the messenger RNA. So the 5' cap is absolutely essential for efficient translation initiation in eukaryotic cells. Now that being said, eukaryotic cells do have eukaryotic transcripts typically, there are some exceptions, typically do have a sequence that resembles the Shine-Delgarno, and in eukaryotic cells it's called the Kozak sequence. In eukaryotic cells, messenger RNAs have a Kozak sequence, which is similar to the Shine-Delgarno in prokaryotes, which is upstream of the start codon, right, the AUG codon, and that Kozak sequence is used to influence the efficiency of translation by interacting with the initiator tRNA in eukaryotic ribosomes. which influences the efficiency of translation by interacting with the eukaryotic initiator tRNA. Okay, number 4. Many, but not all messenger RNAs in eukaryotic cells are polyadenylated on their 3' end. Many, but not all eukaryotic messenger RNAs are polyadenylated on their 3' end. Okay? The presence of the poly A tail, this polyadenylation sequence, helps to increase the lifetime of the messenger RNA so it sticks around in the cell longer before it is degraded. The purpose of the poly A tail is to increase the lifespan of the messenger RNA so the cell can use it longer to make protein. Okay, another point that I want to go back up to number 2 about the cap. Okay, the 5' cap is not only important for translation initiation. The 5' cap, you recall that the splicing of the messenger RNA in a eukaryotic cell is going to take place in the nucleus. Okay, after that splicing is done, that mature messenger RNA has to be removed from the nucleus so that it can find a ribosome to do the translation. Well, the 5' cap is also involved in the proper export of the mature RNA from the nucleus. Five' cap is also involved in the proper export of mature RNA from the nucleus. Okay, number 5. Okay, I think we'll end pretty much on time. I'd ask you to give me a minute or two. Okay, in eukaryotes, there is no need for Fmet. Okay, Fmet is a prokaryotic starting amino acid. There is no need for that in eukaryotic cells. Eukaryotic cells do not use Fmet. Okay.
Excuse me.
Yes.
What did the asterisk say again at the top?
Oh, I'm sorry. There we go.
Proper export of the mature messenger. Okay.
Okay. All right. In eukaryotic cells, Fmet is not used. In eukaryotic cells, Fmet is not used. Okay. And then let me just mention one final thing, which is certainly not exclusive to eukaryotic cells, but it is much more abundant in eukaryotic cells than it is in prokaryotic cells. And that is the fact that many eukaryotic polypeptides go through what's called post-translational modification. That is much more robust, abundant, and commonly found in eukaryotic cells. Okay? Many eukaryotic polypeptides go through extensive post-translational modifications, more so than in prokaryotic cells. Okay, so gang, if you can give me just 2, 3, 4, 5 minutes to finish this up, we'll take a small break. And then I really would like to cover during the virtual office hour, if you can stick around, I really would like to cover your DNA purification work, particularly the calculation of the amount of DNA you isolated from the cells, because I don't think anybody got that right. So if you're able to stick around, that would be very helpful. So let's finish this out. I just wanted to try to give you a feel for what this post-translational modification stuff was all about. Okay, and certainly this could be a known lecture by itself. Your book points out that the scientific community over the years has identified more than 200 different versions, more than 200 different ways that a polypeptide can be modified by a cell. Science has cataloged more than 200 different post-translational modifications. Now, we're not even going to attempt to touch all 200 of those, but I thought you might be interested in knowing a little bit about one thing that I know we all have a vested interest in. And so let me just mention to you the example of insulin, because we all rely on insulin to help keep us healthy. And we know about insulin because of people that struggle with diabetes. Diabetes is a really tough disease to manage. And it's really hard on your body. But at any rate, if I had more time, I'd try to tease it out of you. But you might be aware of the fact that insulin, when it's first translated as a protein, okay, it is translated as a polypeptide. Remember, a polypeptide is a sequence of amino acids that has not yet folded into the proper three-dimensional shape. So the difference between a polypeptide and a protein is that a protein has a three-dimensional shape and a polypeptide not so much. Okay, so insulin is made into a polypeptide, and when it's first made, it's made into a polypeptide that has 110 amino acids. Okay, when insulin is first made, it is made of 110 amino acids. Okay, and in this condition, insulin is actually called pre-pro-insulin. Pre-pro-insulin. Okay, now don't worry too much about this, but I'll just tell you because, well, this is a class. You have these prefixes here, pre and pro. For those of you that have studied any biochemistry and any enzyme biology of any kind, the word pre is always put before a molecule where scientists have realized that the molecule is going to be secreted from the cell. And of course, insulin is eventually secreted from pancreatic cells because it has to get into the bloodstream. Okay, so pre refers to secretion. Okay, and pro, the prefix pro simply tells you when you're reading it in your book that the polypeptide does not have any, I guess I should say, does not have any biological activity at this point. It's kind of sitting there, but it's not doing anything. Okay. So this refers to an inactive status. So here we have a sequence of 110 amino acids that is inactive and eventually will get exported from the cell. Okay. Well, what has to happen next is that this pre-pro insulin is going to be modified by enzymes that are in the cell. Most of these enzymes, I think, modify the protein, the pre-pro insulin in the endoplasmic reticulum. Okay, so the pre-pro insulin is modified, okay, post-translational modification here. It's modified in the endoplasmic reticulum to a sequence of 86 amino acids. And when that processing has completed its work, we now go from pre-proinsulin to simply proinsulin. In other words, the pre part of it has been cut off by the activity of the enzymes in the endoplasmic reticulum. Okay, and then there, excuse me, there are some disulfide bridges. If you remember disulfide bridges from your basic chemistry days, or sulfur bridges, sulfur bonds, there are some disulfide bridges that are linked into place into the proinsulin. And then an additional segment of that proinsulin is again lopped off. To make a polypeptide of 51 amino acids. This sequence of 51 amino acids now folds into the proper three-dimensional shape, is formally secreted by a pancreatic cell, and has biological activity. This 51 amino acid polypeptide folds into a 3D shape and now has proper physiological activity. Okay, so my point here in showing you this is that, you know, when we talk about post-translational modifications, people are so happy to simply have been through the translation process and kind of understand it that they're like, oh, okay, there's this extra thing that, yeah, it's there, but we can kind of ignore it. Well, in the real body, it's not ignored. It can be quite extensive. The chemistry that occurs after translation can be quite extensive. And in this case of insulin, you can see that the initial starting product of insulin is literally cut in half from 110 to 51 amino acids. So there's multiple steps of processing that occur so that your body has physiologically correct insulin to use to regulate blood sugar levels. Okay, I'm done talking to you, gang. Do you have any questions at this time? Okay. All right. I hope you're not passed out on the floor someplace. Let me stop. Is it okay to stop sharing these notes? Have you been able to finish the note taking? Okay. Hayden, are you okay? Okay, Hayden's passed out, I guess. All right, I'm going to stop sharing my screen here. Okay, gang, let's take a 10-minute break. If you're able to stick around, I'd appreciate that. I just want to show you some things about that calculation. And then after that, if there's nothing, you can head off onto your own. So I will be back with you at 2:05 just to talk a little bit about the DNA purification lab. So see you in 10 minutes.
Can you go back to the doc while you're gone?
Okay. Is that what you need, Hayden?
Thank you. Yeah.
Okay, be back in 10 minutes. Okay, gang. For those that were able to stick around, what I wanted to do is go over the DNA isolation calculation with you, because many of you had some trouble with that. Okay, so bear with me just a moment where I get my notes out. So if you do have the lab protocol for the DNA isolation and the work that you did and submitted to me digitally through the Mathematica document, go ahead and get that out. So I want to show you how to do the calculation properly. Many of you were too low in your estimate of the DNA calculation. Okay, so it looks like it's just you and me, Kevin. Okay. All right. Oh, Jose's coming back. Okay. All right, gang. So for the DNA isolation, you had to calculate question F, which was how many micrograms of total DNA did you purify from your starting sample? Okay, there's a couple things that I want to write down for you to frame the context of how you need to do this calculation. Okay, so let's see here. I guess maybe I will go, oh, here comes Malachi. Okay, so what I'll do is go back to the Word document and just type in a couple statements about the DNA purification lab to keep in your mind so we can do the calculation. Okay, so hold on, here we go. Okay, so this will be follow-up on DNA isolation lab. Okay, so for this one, you have to remember a couple things in order to do the calculation correctly. For question F, you need to remember a few key points. Okay, first off, okay, from the standard curve that you folks did, and most everybody got the standard curve pretty well done. I mean, that was not the problem, okay? From the standard curve, you were to estimate the DNA concentration of your DNA sample from your bacterial cells, and that was in tube 6, okay? From the standard curve, tube 6 being your DNA sample, the one that you processed, had an estimated, okay, from the standard curve, and you know, it's okay if I did see a range of values on this, and the value you would have estimated from the curve is in fact just that. It's an estimate. Okay, so there's going to be a window of correctness here. So from the standard curve, tube 6 would have had a DNA concentration around 1.45 to 1.55 milligrams of DNA per milliliter of solution. Okay, and so let's see here. I did my calculation with a concentration of 1.48 milligrams per milliliter. And of course, you recall, as long as we keep the units consistent, that's going to be the same thing as 1.48 micrograms per microliter. It's the same thing. Okay, so most of you did estimate your DNA concentration in that 1.45 to 1.55 range just fine. No problem here. Okay. Now, the next thing to keep in mind is to make your to create your standard curve, you had to take a spectrophotometer reading of your sample. Now, I know we didn't actually do that, and everything you did in lab was sort of a thought experiment. But if you would have done this, okay, the volume of DNA that you eluted off your column, the volume of DNA you eluted off the column that had the DNA bound to it was 150 microliters, okay? The volume of DNA you eluted off the binding column was 150 microliters, okay? That 150 microliters theoretically contains all of the DNA from your starting material. It contains all of the DNA from the bacterial cells that you pelleted at the beginning of the protocol. Okay, this 150 microliters of DNA, excuse me, 150 microliters of liquid contains all the DNA from your starting material. Okay, let me see here. Let me get the chat window up here. Okay, all right. Okay, so by all means, gang, if there's questions or if you're not following along, go ahead and speak up. All right, next. To measure the DNA, you took a portion of that 150 microliters and you would have measured it, you would have measured its concentration in a spectrophotometer, okay? So to measure the DNA concentration, using the spectrophotometer, you took 100 microliters of your eluted sample and you placed it into a tube containing 2 milliliters of distilled water. Okay. So, you know, it's not a big deal at this point because, you know, we're doing things after the fact. But let's see here. If you look at the standard curve protocol in the packet of information I gave you, notice that step number 2 said place 2 milliliters of distilled water in tubes 2 through 6. And notice tube 6 is your purified sample of DNA. So you took 100 microliters of your DNA and you dropped it into a tube with 2 mils of distilled water. So what I'm trying to get you to see from this point is that that means the measurement you took on the spectrophotometer was actually diluted. Okay? This means your measured sample on the spec is diluted. And that's key to doing this calculation. Okay. So gang, question coming through the chat window. Are you with me? Does this make sense? I know it's been quite a long time. It's been many weeks now since we've actually done this. And to really understand it, you kind of got to get your brain back into what you were doing. But to do the calculation, you need to appreciate that the sample of DNA you would have used in the spectrophotometer was in fact diluted because you put it into a tube with 2 mils of water.
So where exactly are you getting the 150 microliters from? Because I see in step 7, it says we took 100 microliters of our purified purified sample and put it into tube 6.
You are correct, Kevin. I'm not at this time telling you you need the 150 microliters necessarily for the calculation. I'm just trying to point out some features of what you did. For example, on the bacterial genomic DNA isolation protocol, notice that step 11 says you put 150 microliters of elution solution onto that column. So that's where the 150 microliters is coming from. In other words, you put 150 microliters of elution solution onto that filter disc in the column, and you centrifuge the column, and the DNA came out in that 150 microliters of liquid. So your purified DNA is in a total volume of 150 microliters at that time. Does that make sense?
Yeah, I got it now. I was just, I guess, not really sure which one to start with.
Okay, fair enough. The reality of it is we kind of need both of these numbers. That's why I'm bringing it up here with the key points that I'm trying to make. So just bear with me a little bit. I think I can get you home here. Okay, Malachi, Ian, are you guys okay? Jose, you okay? All right. Okay. So if you can appreciate these talking points, if you can appreciate these beginning statements that I'm trying to get to you, now we can do the calculation. And so, okay, what I will do here is try to stay on the Word document because it's simply prettier and easier for you to interpret. So the calculation to calculate the answer to question F is going to look like this. Okay, what I'm going to do is put a letter by a number so that I can refer to the letter so you know what I'm talking about. I can refer to the letter so you know which number I'm talking about. So the first part of the calculation is you're going to take 1.48 micrograms per microliter, and that was the value that you estimated from your standard curve, and you're going to multiply it by a dilution factor of 21. Okay, so 1.48 micrograms per microliter times 21. I will come back to that, but okay, so let me define these things for you. A is your estimated DNA concentration from the standard curve. B is the dilution factor. And to calculate the dilution factor, you do this. The dilution factor is equal to the final volume divided by the aliquot volume. Okay, I'll try to explain that better. In other words, for our calculation, you took 100 microliters of your 150 microliters DNA and you put it into a tube with 2 mils of water. That means the final volume was 2,100 microliters. That was going to be divided by the amount of DNA solution you put into the 2 mils of water, which is 100 microliters. So that's how I get a dilution factor of 21. Gang, are you with me? Okay, I hope so. It's really hard teaching this stuff online. I wish we could be in person. All right, so you take 1.48 micrograms per microliter, which is your DNA estimate from your standard curve. You multiply it by your dilution factor, which is the final volume, 2,100 microliters, because that's 2 milliliters, 2,000 microliters of water plus the 100 microliters of DNA solution you put into it. Divide that by the 100 microliters, your aliquot volume of DNA. It's a dilution factor of 21. When you do that, you're going to get a value of 31, excuse me, I'll need another letter here. You're going to get a value of 31.08 micrograms per microliter. Okay, so letter C, the 31.08 microliters is your undiluted DNA concentration because you now have taken into account your dilution factor. Okay, so let me go down here. Letter C is your undiluted DNA concentration. Now, your undiluted DNA concentration is 31.08 micrograms per microliter. And the total amount, the total volume of DNA containing all the DNA from your starting sample is in what volume of liquid, Kevin?
Is that the 100 mics
Not 100.
or the 150?
That's the 150. Good. That's why this gets a little hairy. Okay, so letter D here is 150 microliters. Okay, the 150 microliters is the total volume of eluted DNA from the column. Okay. I'm going to run off the screen here, so let me just do it this way. Letter D down here is the total volume of eluted DNA solution from the purification column. All right, so if we do that calculation, you notice I have microliters on the top for 150 and microliters on the bottom for the letter C number. That means those units will cancel out. And what I'm going to end up with is a value of 4,662 micrograms. That is the total amount of DNA that you in fact purified from your starting sample. All right. Okay. All right, gang. So the problem that I did see with most folks is that you completely skip the dilution factor. Okay, gang. So when you look at your work that you have in front of you, and I know everybody has a little bit of different workflow and setup, do you see how there was at least one critical piece that you missed in your calculation? All right. I don't want to beat a dead horse, but do you have any questions about this? You know, let's be honest, this is not going to show up on your test, so it doesn't kind of matter that much from your current course perspective. But, down the road, depending on what you get into, you're going to have to be able to do calculations like this and make sure that they're right. Okay. All right, so I'll stop sharing my screen here and simply open the floor to you folks. Guys, do you have any questions right now about any course content or anything that you need help with? Will the exam be proctored, Malachi? No, not in any tangible way. You may have heard of a proctoring service online called Examity. Actually, Examity is shut down at this time, so even if we wanted to use it, we can't use it. So your exam is a week away. Next Thursday is your test day. You will log into the ClassMarker website and take your exam at your own home or wherever you're accessing the internet. It's not going to be proctored. Nobody's going to be standing over your shoulder, which is my way of saying I understand that. There's nothing I can do from stopping you looking in your own book, your textbook and notes while you're taking the exam. There's nothing I can do about that. And so I'm okay if you do that. I want you to know it's okay to look in your book during the exam. Nothing bad's going to happen if you do that. But the exam will still be timed. I'm still going to time, I'm probably going to give you an hour and 25 to an hour and 30 minutes to take this test. And once you start it, you cannot stop it. You got to finish the whole way through. So it won't be proctored, but it will be timed. Jose, is the test at the same hour that we have class? No. Next Thursday, when you have your exam, there will be no lecture. There's no lecture next Thursday on a test day. And you will be able to take the test anytime from 7 a.m. to midnight. Any other questions? So, honestly, in some ways, the exam might be a little easier than it normally would be because you can have your textbook and notebook sitting right next to you. But please don't underestimate this. I have some pretty challenging essay questions on here that you're really going to have to think about. And what I mean is you should definitely have some scrap paper nearby you so that you can quickly write down information and work out the logic of problems, because you're going to have to submit a correct answer. And you want to be prepared to think about things in that 1 hour and 30-minute window that I'm going to give you to take the test. Okay, anything else, gang? If not, then, we're good here. I will stay online for a few more minutes if you need help, but otherwise, you're free to log out. And, I wish you continued health and safety to you and your families. We're living under very, very challenging times. So be well, guys.